Well, good afternoon, good morning, good evening, wherever you are. Uh, Welcome to MycoTalks from a very sunny and warm southwest of the UK. I hope it's lovely where you are too. So today we have a great treat. We have two colleagues who are both from Duke. We don't usually have two people from the same institution on the same MycoTalks, but this has happened because of some reshuffling that we've had to do with the availability for various speakers. So we have today Andy Alspaugh and Erica Washington, both from Duke University. And we're really looking forward to two exciting talks. And I'll introduce Andy as our first speaker in a minute. So if we have the next slide, I just want to point out that the MycoTalks has been running pretty much since lockdown and the pandemic. And the idea is to give a very open forum, a global forum for people of all career stages, wherever you are, to give two 30-minute talks and then to use those to participate some in-depth discussion by questions which are submitted by you. And can I just remind you as ever, if you perhaps you haven't attended one before, that you're invited to use the Q&A uh, prompt at the bottom of your screen. Please don't use the chat, use the Q&A, and those questions will be sent to us, and then my co-host, Christina Kumo, and myself will be able to access those and ask questions after we've heard Erica's talk, the second of these two talks. So at the end of this, you'll be able to come back and look at this again if there's something you wanted to see, or recommend this to somebody else to come have a look at it at our MRC Centre for Medical Mycology YouTube channel. So the next slide, please. Thank you. And while we've got you and whetted your appetite, on the 24th, you'll be able to hear on the same channel Jamie Konopka and Jane Usher. And then after that, on the 25th of September, the next month after that, Angie Jelly and Tuo Wang. So two more fabulous talks to look forward to. And also, just to remember, if you're more clinically inclined, I would like to just point out mycoclinics to you. This is where we have more clinical people meeting and discussing, for example, case histories and observations. And that's also been a very, very useful forum for people who are dealing with patient care. So thanks very much for that. And then I've got the honour of introducing our first speaker, Andy Alspaugh. And as you know already, he's from Duke. Andy's one of our physician scientists, or clinical academics, we'd call them in the UK. And his real interest is in the molecular pathogenesis of human fungal infections. He's also very much involved in clinical training in microbiology and infectious diseases, and professional clinical development of graduate students and postdocs and junior faculty. So he's, as I said, an adult infectious diseases physician. He's also actively involved in the training of infectious disease clinical fellows, and especially those who've got an aspiration to go on to be, like himself, a physician scientist. At the Duke School of Medicine, he's been the director of the first year medical microbiology course for 8 years, so he inspires people at all levels. And he's also co-director of the Tri-Institutional Molecular Mycology and Pathogenics Training Program. And he also participates in the long-running course at the MBL, the molecular mycology course, which must have trained generations by now of researchers across the world, and many of the audience may have participated in that. But as I said, what he does in the lab is really focus on molecular pathogenesis, and particularly in infections which occur in the most vulnerable patient communities, especially those with severe defects in immunity, of course, who are particularly exquisitely sensitive to fungal infections. And he's defined in a very specific ways the way in which the fungi sort of sense and then respond to the stress signals to enable them to survive in the infective host. And that leads to his title, which will attract the interest of everybody, dealing with stress and what we learn from a fungal pathogen, lessons from a fungal pathogen. So over to you, Andy. We're very much looking forward to your talk.
Great. Neil, thank you so much. What an honor to be able to speak in this forum. And as Neil said, I'm going to be talking a little bit about stress in microorganisms that cause human diseases. But I just wanted to begin with a story that first caught my attention when I was early in my training as a physician scientist. And I met a man, a 59-year-old farmer. And if anyone recognizes the field, the crop in this field, it's very common in the area around where Duke is, and this is tobacco. And he also, since a young childhood, he was a smoker. And that becomes important because he came to medical attention after not seeing a doctor for decades with the following symptoms: fatigue, weight loss, confusion. And what really brought him to the hospital was he started to cough up blood. And some imaging studies that were done at the time, if you see this on the left is a CT scan of his chest, where you see this mass-like lesion here in the left lung, and likely explaining his bloody sputum, but also in the brain. He had this ring-enhancing lesion at the base of his brain, concerning for, again, a primary lung cancer with metastasis to the brain. During the diagnostic evaluation, fortunately, cancer cells were not seen, but instead they were encapsulated yeasts that were seen by bronchoscopy, and the culture grew Cryptococcus neoformans. And I raise this case for a couple of reasons. It has some typical features of cryptococcal disease, having disease in both the lungs and the brain, but many atypical features. And when many of my microbe focus colleagues hear this case, they ask, "Oh, is this Cryptococcus gattii, something that causes a cryptococcal mass lesions rather than diffuse meningoencephalitis more commonly?" They often ask, was the genome sequenced? Were there some genomic features or phenotypic uniqueness to this organism that would have caused this unusual presentation? Those colleagues who are more host-focused, they first ask, well, did the patient have HIV infection, other defined immunosuppression? Did he have antibodies against GM-CSF or other cytokines that would have put him at risk for this infection? Many of these things are going to remain unanswered. We don't have the organism anymore, and the patient has long since expired. However, as this organism entered into this patient from the environment, it had to go from a unique set of stresses in the external world to a different set of stresses as it entered into his body. And many of these stresses would have included varying temperatures, solar radiation, and nutrient deprivation, natural predators. Some of these would be similar to stresses that the patient would have that the organism would have experienced in the host. Variations in temperature and pH, living within and outside immune cells, reactive oxygen nitrogen species, metal, and other nutrient deprivations, and just many aspects of the host immunity. What I'd like to do is I really want to focus on one of these stresses, and it's the metal stress. And specifically, I'd like to talk about some of the adaptations that this organism, this particular species, will try to accomplish in order to survive within the host. So many people on this call will have studied morphological transitions within fungi, such as titanization, that will allow the organism to survive in a very hardy state in the setting of the immune system. Cryptococcus produces, as many of you know, a polysaccharide capsule that protects it from the immune system. It also changes its cell wall dynamically in response to varying conditions, including the host. And we'll be talking a lot about the cell wall today. The organism must also adapt to the temperature of the host. I'm not sure that the external and internal temperatures are any different in North Carolina today, but typically the host is much warmer than the environment. And I'd like to really specifically focus about copper. There are some unique aspects of copper biology and pathogenesis. As many of you know, copper because of its redox potential, is able to cycle between a copper 1 and copper 2 state, and it really drives the activity of many enzymes, such as those required for iron import, those involved in oxidative phosphorylation, also for scavenging of reactive oxygen and other cell-damaging species. And in Cryptococcus, the LAC1 enzyme is a copper-dependent enzyme that drives melanin production. And because of that, the host will employ strategies that will try to specifically withdraw copper from invading pathogens in a concept known as nutritional immunity, and really driving copper levels so low that it may compromise some of these essential copper functions. But a different aspect of nutritional immunity that is not as appreciated, but is really important with copper, is the concept that in specific micro niches within the domain, the copper levels are actually incredibly elevated. The host will bombard the pathogen with copper in order to induce direct cellular damage. Copper can replace other metals and cause mismetallation of enzymes. And again, because of its redox potential, can also induce oxidative damage to different macromolecules within the cell. So within the host, and I'm sorry, so this potential for copper bombardment has been used therapeutically. Copper was used as an antimicrobial agent back in ancient Egypt, and it's still being used today, both in medical and agricultural applications to try to kill fungi. This is relevant in the context of host infection. So in different microniches, as we said before, the host will actively sequester copper away from the microorganism, and particularly in the human brain, the copper levels are very low, and causing Cryptococcus to induce a set of low copper genes, such as those involved in the effective importation of copper into the cell. And if these genes, if the 2 copper importers, for instance, are mutated, then we see a dramatic decrease in the ability of the organism to maintain an infection at this site. In contrast, in the lungs, there are places within the lungs where the organism will experience a high copper state, that copper bombardment, requiring a completely different set of genes to be induced to protect it from this unique copper environment, including MT1 and MT2 metallothionein genes. These proteins are copper sponges that really take copper away in the presence of copper toxicity. And again, if you mutate these 2 metallothionein genes, again, and infect mice intranasally and by inhalation, we see a dramatic decrease in the ability of the organism to cause disease. Interestingly, there is one transcription factor, the CUF1 protein, that mediates both the low copper gene response and the high copper gene response. And Peter Williamson demonstrated that in a systemic model of cryptococcus that a deletion of the CUF1 gene resulted in a dramatic decrease in the ability of the organism to cause disease after intravenous infection, really further emphasizing the importance of proper copper homeostasis in this organism. And so at Duke, there were a series of investigators led by Dennis Thiel that looked at this one copper transcription factor, CUF1, and how it controlled responses both high and low copper levels. Again, in low copper levels, you can see genes induced involved in copper importation. And in contrast, copper detoxification machinery was induced in the presence of toxic copper levels. And these experiments were done by both RNA-seq, comparative transcriptional profiling, as well as ChIP-seq, looking at those specific promoters that were targeted by this CUF1 transcription factor. So I really want to tell 3 brief stories today. First, how some of the work that we have subsequently done has looked at different models of copper acquisition. Also, how maybe copper-dependent enzymes, both those that are transcriptionally controlled by copper and those that contain copper, are involved in cell wall adaptations to different stress conditions. And then last, how can one transcription factor identify and bind both low and high copper genes? So first, let's talk about copper acquisition. As I mentioned before, it was pretty obvious that copper transporters such as the CTR1 and CTR4 would be induced in the setting of a low copper state in order to make sure that the cell had adequate copper resources. But there were other genes that were less apparent. And the one I want to focus on is a CBI1 gene that was co-transcriptionally regulated with the copper importers. And so the question was, what does the CBI1 protein do? So both Dr. Probst and Dr. Garcia Santamarina have worked on this and first noted that the CBI1 mutant had a specific defect in the ability to grow in the presence of copper limitation. And the limitation was induced by this copper chelator, BCS. So the higher concentrations of BCS, the less copper available. And you can see that the wild type strain can grow up into a certain amount of copper deprivation, but both a copper importer mutant as well as a CBI1 mutant were unable to grow in the presence of low copper states. And in contrast, the copper sponges are not required for growth in the presence of low copper, but in high copper. Additionally, in addition to its inability to grow at low copper, there are some unexpected phenotypes in the CUF1 mutant and in the CBI1 mutant where they were exquisitely sensitive to the addition of compounds into the medium that would stress the cell surface. Things like sodium chloride, SCS, Conger red, chalcofluor white, and you can see their attenuated growth compared to the wild type or complemented strains for cell wall stressors. So what does CBI1 do? Well, just looking at the protein itself, it resembled somewhat by sequence to this set of enzymes called LPMOs, or lytic polysaccharide monooxygenases. Interestingly, these are copper-containing enzymes that serve to make nicks in crystalline polysaccharides. You see this planar surface here. They sit on a planar sheet of polysaccharides, and they create holes or nicks in it to allow specific sugar hydrolases to better penetrate and have activity. So you can imagine in the environment, this may be important for breaking down cellulose in the environment too. But also the cell surface, the cell wall contains these types of complexly coordinated polysaccharides that may actually be modified by things like these LPMOs. In collaboration with Katja Johansson at the University of Copenhagen, who is an expert in this set of enzymes, they realized that, in fact, there was no documented LPMO enzymatic activity in the CBI1 protein. However, in response to copper, in response to different redox stresses, the copper didn't stay in the enzyme and fluctuate between states. That it was actually bound or released based on the oxidative stress, which is very different than these true LPMO enzymes that kept the copper bound well in the active site. So later on, Dr. Probst and Dr. Garcia Santamarina determined that CBI1 likely had a very different function. This binding and releasing of copper in response to stresses seemed to indicate that perhaps the CBI1 protein would bind copper under certain conditions. And then also noted that it physically associated with the copper importers. And so perhaps it would release copper and allow it to be imported under other conditions. So some of the evidence that would support this. So we believe that this protein exists within in the cell surface. And if you look at the cell wall, this is a transmission electron micrograph of the wild type strain grown in copper sufficiency and in copper deficiency. And you can see a difference in the electron density of this structure, likely indicative that copper is being deposited within the cell wall in this condition, and it's being removed from the cell wall in this condition. And in contrast, in the CBI1 mutant, there's no significant change in the electron density of the cell wall, suggesting that perhaps if this is copper causing the signal, it is not being mobilized in the absence of this protein. Well, this would make sense that the cell wall would bind copper if it was known that cell wall components actually did bind copper. Well, that is known. So this is looking at a chitin, and then a related molecule, chitosan, that is related to chitin, except it is the deacetylated form of chitin. And this deacetylation actually opens up this amino group that allows copper to bind to this polymer. So chitosan is a very, very avid binder of copper, and has actually been used in environmental bioremediation experiments where it can remove it. And so the hypothesis is that chitosan serves as a storage site for copper. The cell wall serves as a storage site for copper during copper excess, and it mobilizes copper during copper limitation, but it could also potentially serve to protect the cell from copper toxicity. We see in the CBI1 mutant that specific cell wall genes are altered in their regulation, including the CDA2 chitin deacetylase that's involved in the conversion of chitin to chitosan. And we see by biochemical measurements that there is a decreased amount of total chitin and chitosan in the CBI1 mutant strain. And this results in some changes that we see in the cell wall organization, as noted here by the WGA staining, which we typically only see little foci of exposed chitin in the wild-type strain, but a much increased exposure of chitin and chitosan in the CBI1 mutant, indicating a significant alteration in its organization. Also, if we treat the CBI1 mutants with echinocandins, we see a dramatic increase in the activity of echinocandins to inhibit the growth of this fungus. As many of you know, echinocandins have a little primary activity against this fungus, but when we mutate CBI1 and we change the cell wall structure, we see an enhanced susceptibility to echinocandins. So it seems to be that the cell wall and chitosan are required for copper storage. Does it affect copper stress resistance? Well, if we mutate genes involved in chitosan production, such as the chitin deacetylases, CDA1, 2, and 3, Jenny Lodge in her lab several years ago demonstrated that if you mutate all of these, we had almost no detectable chitosan in the cell wall of the strain. And in a CDA 1, 2, 3 triple mutant, we see a dramatic increased susceptibility to the addition of copper to the medium, similar to a strain that no longer encodes the metallothionein copper sponges. Also, if we mutate the gene encoding a chitin synthase that's responsible for the chitin that's converted to chitosan, it has a similar decreased growth in the presence of exogenous copper. So it seems that chitosan itself is important for copper stress resistance. So in this part of the talk, there's this dynamic between copper homeostasis and the cell surface composition and architecture, where the cell wall can likely serve as a protective barrier, a binder of copper during copper excess, but also potentially as a storage site for copper during periods of copper limitation. Just to transition to a different, so what about copper enzymes and mediating different cell wall adaptations? I'd like to return to this concept of these LPMOs, because LPMOs are a really important and understudied group of enzymes in the context of fungal pathogenesis. Again, I mentioned they create little nicks or areas of access within a complex polysaccharide that allows degrading enzymes better access and to better break down complex polysaccharides. And they serve many roles in nature, again, including breaking down cellulose and other polysaccharides, and they're used in industrial purposes. Well, there are 4 LPMO or LPMO-like encoding genes in Cryptococcus 1, CBI1, which I indicated is not a true LPMO. But there are others that look more like these LPMOs, including this one, AA9. I really want to focus on it and think of what might it be doing. Is it being secreted in the external environment and degrading plant material, or is it doing something more involved with the organism itself? Again, it's a predicted group of this carbohydrate active enzyme referred to as AA9s, which typically break down cellulose. But it's interesting, when we look at established transcriptional databases, it's actually highly induced in the setting of infection, that and one of the other LPMOs as well. It's also induced specifically by an alkaline pH signal, and I'll sort of show that again here. And it's required for growth under tissue culture conditions, 37 degrees, 5% CO2, that mimic the host environment. I mentioned that it's induced, the gene is induced, but this is a Western blot showing the induction of the protein specifically at pH 8, and not by any particular copper condition. And we see that this induction of the protein is dependent on the alkaline response pathway transcription factor RIM101, in which we do not see any protein even at otherwise inducing conditions. And there is also a slight dependence on temperature for the induction of this protein. So two conditions that would be relevant for the host. When we actually looked at the importance for cell one in the context of pathogenesis, we saw that the mutant was absolutely virulent in a inhalational model of Cryptococcus compared to both the wild type and the complemented strain. We mentioned that it didn't cause disease, but also the organism just was unable to establish an infection. These are the colony-forming unit counts from the lungs at several different times after infection, and where we see an increase in the number of colonies in the wild type infected mice, we see stable or decreasing amount of the organism to be able to survive within the host environment. So what's the function here? Well, we by several experiments, we were able to demonstrate that this protein is not being secreted into the external environment. We don't see it in the supernatant. We don't see it in sort of cytosolic fractions, but instead we see it in the cell pellet, likely associated with the cell surface. And as we degrade the cell surface with zymolyase, we see a decrease protein captured, suggesting that it is when we digest the cell wall, we actually liberate this protein from the cell itself, suggesting its cell wall localization. Well, if it's not being secreted into the environment, is it having an effect on the cell wall? And we were informed by some data from a bacterial Streptomyces species, in which an LPMO in that species was able to modulate the cell wall in response to different stresses, nicking the complex polysaccharides and allowing hydrolases to remodel the cell wall. So our model was that perhaps that's what was happening in Cryptococcus, the dramatic cell wall remodeling that happens in response to host stresses perhaps was facilitated by this LPMO. Well, in a similar set of experiments where we look at different cell wall stresses, at 30 degrees on rich medium, a condition in which this protein is not expressed, we don't see any significant alterations in cell wall stresses. In contrast, as we look in conditions in which the organism would, and the protein would be expressed, we see a significant susceptibility to alterations in pH or other cell wall stresses. Now, also, if we just increase the pH and don't change the temperature, we also see increased susceptibility compared to wild type of the cell one mutant for different cell wall stresses. Just looking at the cell wall itself by wheat germ agglutinin and calcofluor white staining, we don't see significant alterations in total chitin or exposed chitin activity. But what we began to see is at 37 degrees, some alteration in the morphology and the budding patterns of this strain. Cell wall content is reasonably intact, but we do see a slight increase in beta-glucan levels within this strain. But if we look at the morphology of the strain, we see that they're quite monomorphic. We don't see the range of budding that we do in the wild type strain. This can be seen both by ND ink stain or by scanning EM, looking at the size and some of the capsule of the strain. And we also see an increase in the total cell body size and a change in the exopolysaccharide, the secreted capsule, suggesting that perhaps it is having some kind of effect either on the cell wall and the types of capsule that's secreted, or perhaps on the capsule itself. Thinking about the alterations in cell size, the question was, is there some kind of cell cycle defect? And indeed, we see alterations in the budding pattern as well as alterations in ploidy. So under conditions in which this protein would be expected to be expressed, we see that the ploidy looks more like a stable diploid rather than the wild type strain, suggesting that this protein is involved in the progression of this organism through the cell cycle, and the absence is perhaps arresting under this non-permissive growth condition in this unbudded G2 state. So just in 1 minute, I'd just like to just sort of highlight some experiments we're doing now to look at how this one transcription factor, CUF1, can actually interact with some of these copper-regulated promoters and regulate both a copper response to copper toxicity and to copper deprivation. In other fungi, there are two copper regulating transcription factors, one called MAC1 that induces genes or regulates genes in response to low copper state, and ACE1 that regulates genes in response to a high copper state. And in contrast, the single CUF1 transcription factor in Cryptococcus seems to have features both ACE1-like and MAC1-like, in addition to its DNA binding domains. So how might this be regulated? Well, Corinna Probst made some mutations within these different sites. There are cysteine-rich sites here that likely mediate copper binding, and she was able to show that just the mutation of this first cis-rich region, this ACE1-like cis-rich region, completely abrogated the ability of the organism to respond to high copper, but did nothing to its ability to respond to low copper. So shown here, you can see that the CUF1 strain that's responsible for both responses cannot grow in copper limitation or in excess copper. And in contrast, when just this one cis1 site is altered, the strain can still grow in copper deprivation, but not in copper toxicity, suggesting that these are some of the functional units within the copper transcription factor that allow it to mediate its effect. And she also demonstrated that this really affects binding to high copper promoters, such as the metallothionein promoters, the ATM1 promoter, but not to low copper promoters, such as CTR4 and CBI1. So she's been able to now significantly tease apart the high copper stress from the high copper response from the low copper response to this transcription factor. And for the sake of time, I just want to indicate that in the setting of infection, this has physiological significance because we see significant alterations in the immune response to the CUF1 strain and the ACE1 mutated strain, in contrast to the wild type strain in terms of the number of fungi and where they're located. The fungi in this strain are all grouped together in these small little foci of inflammation. And so we're really excited to see how the copper response is affecting the host response to this organism. And the next speaker, Erica Washington, is a brilliant biochemist we're working with her to isolate the CUF1 protein and to begin to do some biochemical DNA binding studies to see how the different features of this transcription factor affect its ability to mediate a copper response. So in conclusion, I just want to hopefully have tantalized you with some of the real richness of the copper, the role of copper in microbial pathogenesis, and that it has different effects in different parts of the body. We've demonstrated that there are newer models of copper acquisition that we're able to see that the cell wall is intimately involved with copper homeostasis, and that the different copper enzymes are involved with that. And we're excited to look at different types of copper regulation. And we have in this single strain, Dr. Probst has linked the green fluorescent protein to a promoter that is induced in low copper states, the red fluorescent protein that is induced in high copper states. And we're going to be able to probe different host conditions based on alterations in fluorescent protein expressions. With that, I just want to thank the many people who've been involved in these projects and with our labs over the years. And I look forward to the next talk.
Thank you, Andy, for that very interesting talk. As a reminder, we will have question and answer for both speakers at the end. If you can please post your questions in the Q&A with the name of the speaker that your question is for. I'm pleased now to introduce our second speaker. Dr. Erica Washington is an assistant research professor in the Molecular Genetics and Microbiology Department at Duke University School of Medicine. Dr. Washington has a secondary faculty appointment in the Duke University School of Medicine Biochemistry Department. Dr. Washington has been awarded the Duke Next Generation Fellowship funded by the Duke Science and Technology Initiative. Dr. Washington completed her postdoctoral training as a tri-institutional molecular mycology and pathogenesis training program fellow with Dr. Richard Brennan in the Department of Biochemistry at Duke University School of Medicine. She used X-ray crystallography and cryo-electron microscopy to determine the structures and functions of enzymes in a metabolic pathway that is critical for virulence in fungal pathogens. Dr. Washington completed her doctoral training with Dr. Jeffrey Dangle at the University of North Carolina, Chapel Hill, where she studied the molecular mechanisms by which type 3 effector proteins from the plant pathogen Pseudomonas syringae contribute to pathogenesis. Her work has resulted in landmark publications in the Proceedings of the National Academy of Sciences, Cell Host Microbe, and Cell Reports. Erica, we're very excited to hear your talk. Please share your slides and tell us about how sweet it is.
Thank you. Okay, thank you, Christina, for that kind introduction. And I'm very honored to be able to present my work today in this forum of the MycoTalks seminar series. Okay. Here we go. So as was mentioned, I started the Washington lab at Duke School of Medicine in the Molecular Genetics Department of Microbiology in 2003. And in the Washington lab, we use a combination of structural biology, biochemistry, and fungal genetics to answer questions about fungal stress responses. And then we use what we learned to harness this information to develop new antifungal drugs. And so today I'm excited to share two stories that we've been working on that encompass the multiple facets of our research program. So these two stories start with trehalose. Trehalose is a non-reducing disaccharide made up of two glucose molecules. And trehalose was first identified in the 1600s in these insects called Larinus beetles. And since then, trehalose has been chemically studied and its chemical properties have been studied in multiple organisms. And we know now that trehalose can act as a stabilizer of membranes and proteins. It can act as a carbon source, contributing to energy homeostasis by being broken down into two glucose molecules. And trehalose can also act as a stress protectant. And this includes the ability to protect organisms against dehydration, high salinity, hypoxia, nutrient starvation. And what's important for our talk today is the ability of trehalose to protect organisms against heat stress. And we're interested in understanding how fungi respond to heat stress because as environmental fungi, Fungi living in the environment are exposed to slight increases in temperature due to our warming climate, or even drastic sudden increases in temperature due to our warming climate. This gives the ability of fungi to evolve and gain traits that allow them to survive at higher temperatures. In crossing this disease prevention barrier we call the thermal restriction zone, may allow fungi to become potential pathogens in humans. And so one of the questions we're interested in answering in the Washington lab are, what are the thermal adaptation mechanisms that would be dangerous for these environmental fungi to evolve? And one of those mechanisms is the trehalose biosynthesis pathway. So the canonical trehalose biosynthesis pathway in fungal pathogens starts with the enzyme TPS1. TPS1 is a glycosyltransferase that takes substrates G6P, or glucose 6-phosphate, or UDPG, or uridine diphosphate glucose, and converts these two substrates to trehalose 6-phosphate, or T6P. This releases UDP. The second enzyme in this pathway is TPS2. TPS2 is a phosphatase, and so it removes the phosphate group from T6P, and we get our final product of trehalose. Trehalose can also be broken down by neutral trehalases to generate glucose molecules. So when we look at this biochemically, we can see the UDP glucose as our sugar donor that donates it to the glucose 6-phosphate, allowing the creation of trehalose 6-phosphate and the release of UDP. And then this is the phosphate group that is removed by TPS2, yielding trehalose. Okay, we focus most of our studies to this point on the trehalose biosynthesis pathways in Cryptococcus neoformans and Candida albicans. Cryptococcus neoformans is a deadly basidiomycete that causes meningoencephalitis in immunocompromised patients as well as other patients. Candida albicans is normally part of our normal host microbiome, but it can also cause invasive infections in immunocompromised patients. And so the severe morbidity and mortality that's due to these two pathogens has led them to the World Health Organization list of critical fungal pathogens. And so in these two fungal pathogens, we know that an increase in temperature exposure to heat stress leads to an induced accumulation of trehalose. So here we can see in Cryptococcus neoformans, when wild type cells are exposed to 37 degrees, which is heat stress for these cells, this leads to an induction and accumulation of trehalose. And this is dependent on that first enzyme in the pathway, TPS1. Similarly, in Candida albicans, when wild type cells are exposed to 42 degrees, again, this is their heat stress temperature, you can see an even more drastic accumulation of trehalose, and this is dependent on TPS1. Furthermore, this increase in trehalose accumulation is correlated with an increase in thermotolerance. So when we look at Candida albicans cells, wild type cells are able to grow at 30, 37, and 42 degrees. However, when you knock out the two copies of TPS1, the ability to grow at these higher temperatures is greatly reduced. The same can be said for Cryptococcus neoformans. Wild type cells can grow at 30 and 37 degrees. However, the TPS mutant has the growth at 37 degrees is greatly abrogated. And this thermotolerance is one of the mechanisms and one of the contributions of the trehalose biosynthesis pathway to fungal virulence. And so when we look here in a mouse pulmonary infection model, we can see that mice that are infected with the TPS1 mutants are able to survive 28 days, the course of the experiment. However, this is in contrast to the mice infected with wild type cells that are only able to survive between 22 and 25 days of the experiment. We also have the lung burden has also been looked at in these mice experiments. And so what we can see is when we look at the fungal burden at 1, 3, and 7 days, by 24 hours, there's already a significant decrease in the fungal burden that can be identified for a TPS1 mutant. And by 7 days, the TPS1 mutants are completely eliminated from the host. And this indicates to us that TPS1 mutants are fungicidal. And And so we're interested in targeting the trehalose biosynthesis pathway with an antifungal drug. And we are interested in targeting this pathway because we want to develop a drug that can target current fungal pathogens, as well as environmental fungi that may be thermotolerant and have the potential to become pathogens of humans. As an antifungal drug target, the trehalose biosynthesis pathway has really important properties. As I've just shown you, these mutants are fungicidal, so they can't survive in a host. Our structural biology knowledge of these enzymes leads us to the ability to develop a broad spectrum and/or specific antifungal drug. Perhaps most importantly, the trehalose biosynthesis pathway is not found in humans. So this gives us the ability to develop a safe and low toxicity drug that is safe for the immunocompromised patients that are often susceptible to these invasive fungal infections. And so we decided to take a structure-based iterative drug design approach, starting with a high throughput screen, followed by structure-guided optimization. And we'll follow this up with medicinal chemistry, mammalian models of infection, and hopefully clinical trials. And we were able to take this approach because we've developed protocols to now enable us to purify highly pure TPS1 and TPS2 proteins with high yield for screening. We've developed biochemical and biophysical screening assays, as well as determined multiple structures of TPS1 and TPS2 from these fungal pathogens. So some examples of these structures are TPS1, the glycosyl-transferase, that first enzyme in the pathway. There are crystal structures from the Candida albicans TPS1, Alspaugh fumigatus TPS1, and the Magnaportho rhizae rice pathogen TPS1. And what we can see when we look at these structures of TPS1, we can see that this enzyme has two subdomains, an N-terminal domain and a C-terminal domain. And in between these two domains is a deep cleft with modified Rossmann folds for binding of the substrate. So the N-terminus binds G6P, the C-terminus binds UDP glucose, and the two subdomains are connected by C-terminal alpha helix. So what you might notice when you look at this list, however, is that there's no Cryptococcus neoformans TPS1 on this list. And that's one of the pathogens that we're particularly interested in. And that's because we had to use a different method to determine the structure of the Cryptococcus neoformans. So today I'm going to tell you two different stories. First, in part one, I'll tell you about our use of cryo-electron microscopy to determine the Cryptococcus neoformans TPS1 structure. And then part two, I'll tell you about our work to identify a compound that inhibits TPS1 activity. Okay. So the Cryptococcus neoformans TPS1 has a large 92 residue intrinsically disordered domain in the middle of the protein. And this large flexible loop has made it incredibly difficult for us to generate crystals that are well ordered and resolved to a high enough resolution. However, this intrinsically disordered domain adds 20 kilodaltons to the protein compared to the Candida albicans TPS1. We hypothesize that TPS1 forms a higher molecular weight oligomer. And so with 74 kilodaltons, if this forms a higher molecular weight complex, this would be in the range of a size of a protein that we could determine the structure of by using cryo-electron microscopy. And so that's what we decided to seek out to do. So we started with highly purified Cryptococcus neoformans TPS1 full-length protein with an N-terminal histag. We froze down Cryptococcus neoformans on grids, on cryo-electron microscopy grids. And so here you can see these tiny dots are the particles of Cryptococcus neoformans TPS1 on these grids. We were so excited when we saw these particles, and we moved forward from the screening step to do a full data collection on the Titan Krios microscope here at Duke. We collected more than 2,000 micrographs and picked over 1 million particles. And that enabled us to generate 2D class averages where we could see that there was some interesting structure happening in these Cryptococcus neoformans TPS1 particles. Based on the 2D class averages, we generated a structure that was 3.3 angstrom global resolution map. And from this map, I was able to build the model of the Cryptococcus neoformans TPS1 APO full-length protein. So we could see that this protein forms a homotetramer, where there's 4 protomers in this map of the Cryptococcus neoformans TPS1. I started the building of this model using the Canada albicans crystal structure. And unfortunately, many of the flexible regions that were at the N-terminus and the C-terminus, and also the intrinsically disordered domain, were not able to be resolved in this cryo-EM structure. So next we moved on to looking at the structure of Cryptococcus neoformans TPS1 bound to substrates. And our goal here was multiple. We wanted to see if you could get a higher resolution structure. And then we also wanted to see if you could observe a conformational change between APO and ligand bounds. And lastly, we wanted to see if we could detect small molecules in this structure. And that would be a proof of principle that we could use cryo-electron microscopy to study potential drugs that would be bound to the Cryptococcus neoformans TPS1 protein for our drug design work. And so we decided to incubate TPS1 with glucose 6-phosphate, its substrate, and UDP, which is its product. And so we follow the same pipeline of the cryo-electron microscopy, except for this time we had excess of UDP and G6P added to our protein. We, again, had beautiful particles on these grids. We did a data collection, again, had amazing 2D class averages. And this allowed us to solve the structure to a higher resolution of the Cryptococcus neoformans UDP G6P complex. And so what we could see when we built the map model into this map, we could see that we could still see the 4 different protomers in this cryo-EM structure, but inside each of the substrate binding pockets, we were able to identify G6P and UDP. So this was very exciting. We could see high enough resolution in that substrate binding pocket that we could make out specific detailed interactions between substrate binding residues and the ligands. Unfortunately, we still could not detect the structure and to determine the structure of the intrinsically disordered domain. And so we'll have to do additional tricks to figure out what's going on there. But when we look at the substrate binding residues that bind UDP and G6P, we could see specific hydrogen bonds that were formed. For example, this glutamic acid 522 formed 2 hydrogen bonds with these exocyclic hydroxyl groups 2 and 3 on UDP. We could also see interaction between the phosphate group and arginine 453. So I mutated these residues and measured the activity of TPS1 and confirmed that these residues are required for TPS1 activity. Furthermore, these residues are conserved in other fungal pathogens as well as bacteria. So we learned additional things from these structures. We did observe a conformational change. So here in blue, I'm showing you the APO or unbound Cryptococcus neoformans structure. And then in green is the Cryptococcus neoformans TPS1 bound to UDP and G6P. And what you can see is there's movement inwards towards the substrate binding pocket of alpha helix 2. And this is a 7 angstrom movement. It also led to a 29-degree closure and rotation of the substrate binding pocket. We were also able to identify critical interactions in both interfaces of the Cryptococcus neoformans TPS1 homotetramer. So interface 2 and interface 1 and interface 2. So next we wanted to look at the specificity of the Cryptococcus neoformans substrate binding pocket. And for this, we utilized UDP galactose. UDP galactose was a great option for us to use because it's structurally very similar to UDP glucose. On the 4th carbon, there's a change in the position of the hydroxyl group compared to UDP glucose, and that's the only change. So we wanted to see if TPS1 could use UDP galactose as a substrate. So we added the UDP glucose, UDP galactose, and then UDP as a negative control, and again measured TPS1 activity. And what we could see is that UDP galactose is not a substrate for Cryptococcus neoformans TPS1. So then we wanted to see if UDP galactose can bind at all. And so here we did a competition assay and we added in 10-fold excess UDP galactose compared to UDP glucose. And then we could see a 30% reduction in the activity of the relative activity of TPS1, indicating to us that although TPS1 can't use UDP galactose as a substrate, UDP galactose can in fact bind weakly and compete off UDP glucose, the native substrate. Okay, so now we have our structural information. We have crystal structures from the Candida albicans TPS1, Aspergillus TPS1, the cryomium structure of the Cryptococcus neoformans TPS1. So in part 2, I want to tell you about our work to identify a compound that inhibits TPS1. So we started off with a high throughput screen. And this is a fluorescence polarization screen where we're looking at the rotation of a fluorescently tagged UDP molecule to indicate whether or not the activity of TPS1 has been decreased in the presence of compounds. We used highly purified protein. So again, this is internally his-tagged Candida albicans TPS1. And then we started with a screen of 750 compounds. In the second step we found that we had 32 compounds that inhibited the activity of TPS1. Then we did dose response assays and we had a final hit of 7 compounds. And so I'm showing you here in this panel on the right, these 7 compounds are in green, and I've highlighted here in blue SJ6675. And SJ6675 was of interest because it was one of the compounds that we were able to generate a co-crystal structure of with the Candida albicans TPS1. And so we could see that SJ6675 is binding in the substrate binding pocket. Although this is a low resolution structure, we were able to use this as a scaffold to build a library of derivatives of SJ6675. So we took that library and determined if any of these derivatives affect the activity of TPS1. And we were very excited to see that 4456, in fact, inhibits the activity of TPS1. And so we became more excited about pursuing 4456. And we generated a soluble version of this compound, which we now call 4456 dichloride hydrate, or 4456DH. Okay, so we first wanted to determine if 4456DH is able to bind TPS1. And so we did microscale thermophoresis experiments to show binding of the Candida albicans TPS1 and 4456DH, as well as Cryptococcus neoformans TPS1 and 4456DH. And these were micromolar amounts of concentrations of binding, KDs. We also were able to show that 4456 inhibits the activity of Candida albicans TPS1 and Cryptococcus neoformans TPS1. So next, to determine the mode of action of 4456, we again pursued co-crystallization experiments. And with this experiment, we were able to see that 4456 does in fact bind in that deep cavity substrate binding pocket of Candida albicans TPS1. And in fact, we could also see that there was closure of that alpha helix 2, kind of burying and closing the enzyme in on 4456 and that substrate binding pocket. So the position of 4456 in that substrate binding pocket means that it's proximal to substrate binding residues, as well as the place where you would normally see the substrates UDP and G6P. Furthermore, these substrate binding residues that are near 4456 are conserved. And so this gives us an indication that with 4456, we might be able to develop a broad spectrum antifungal drug. So this was great. We had binding We had a mode of action indicating that this is a competitive inhibitor binding in the substrate binding pocket of 4456. But how does 4456 DH affect fungal cells? To answer this question, we did MIC assays, minimum inhibitory concentration assays, to determine if 4456 inhibits the growth of cells. And we were excited to see in the presence of elevated temperatures, so exposing the cells to heat stress, we were able to see that in a concentration-dependent manner that 4456 inhibits the growth of multiple Candida species and Cryptococcus species. Furthermore, we were able to show that 4456 decreases trehalose accumulation in both Candida albicans and Cryptococcus neoformans. So in this assay, we expose cells to either 30 or 42 degrees, and then in the presence or absence of 4456 DH. And so what we could see in the absence of 4456 DH, there was an increase in trehalose as expected and was published previously. But then when we add sub-MIC concentrations of 4456 DH, we see a decrease in trehalose accumulation. And we see the same phenotype in Cryptococcus neoformans, where an exposure to 37 degrees compared to 30 degrees leads to an increase in trehalose accumulation. And this is decreased when you add sub-MIC concentrations of 4456 DH, indicating to us that 4456 is working as expected by targeting TPS1, leading to the decrease in trehalose accumulation under heat stress. Now, we would next wanted to determine if there's any defects in virulence factors that are associated with trehalose biosynthesis. So in Cryptococcus neoformans, it's known that deletion of TPS1 leads to reduced capsule formation. And so here we did a dose response experiment and we looked for increasing doses and their effect on capsule formation. And we were able to see by increasing the concentration of 4456 DH, there was a decrease in capsule formation. Similarly, in Candida albicans, we know that deletion of TPS1 leads to reduced filamentation in Candida albicans. And as a result, we did a dose response assay and looked for, does 4456 DH affect filamentation in Candida albicans? And at these higher concentrations, this treatment of 4456 DH phenocopies the Candida albicans TPS1 double mutant. Again, indicating that 4456 is working by targeting TPS1. So our current working model for 4456 DH is that it targets TPS1. And so here I'm showing TPS1 as its homotetramer form, where 4456 is binding in this highly specific substrate binding pocket. And this leads to a reduction in trehalose, which is induced during heat stress. We're also open to the idea that 4456 DH can target an unknown target, perhaps another glycosyl transferase or enzyme with a similar structure to TPS1 and fungal cells. And so the take-home message that I want to leave you with from these two stories about trehalose biosynthesis and antifungal drug targeting is by targeting the trehalose biosynthesis pathway, we can modulate thermotolerance in emerging fungal pathogens and also reduce the virulence in the critical fungal pathogens that we already have in our clinics today. And so trehalose biosynthesis pathway is a very critical antifungal drug target. So there's still more work to do. This is just the tip of the iceberg. We are interested in moving forward and studying trehalose biosynthesis in environmental and emerging fungal pathogens. As I mentioned earlier, our work has focused on Cryptococcus neoformans and Candida albicans, and these are the wild-type lab strains so far that we've worked on. And so we're really interested in exploring environmental and clinical strains that are less studied to see if there's any differences in trehalose biosynthesis. We're also pursuing suppressor screens to elucidate the mechanism by which TPS1 and TPS2 contribute to thermotolerance. And lastly, we're interested in pursuing environmental evolution approaches and to investigate if evolution in Increased trehalose biosynthesis is sufficient to take an environmental pathogen that's not thermotolerant and to turn it into something that's thermotolerant and a potential pathogen. So with that, I'd like to acknowledge all of the collaborators that I've been so lucky to work with on this project. Richard Brennan, Alberto Bartasaghi, as well as members of Richard Lee's lab at St. Jude Children's Hospital, members of John Perfect's lab at Duke University, and Joe Heitman's lab, also at Duke University. I've also been so fortunate to have amazing faculty mentors, Andy Alspaugh, Jenny Lodge, Amy Gladfelter, and Dennis Combe. And I'd like to thank sources of funding and support, the Duke Next Generation Leaders program, which is funded by the Duke Science and Technology Initiative. I'd like to thank the Advanced Light Source for access to X-ray beams, and the Tri-Institutional Molecular Mycology Pathogenesis Training Program that gave me support and supported me during my postdoc. I'd like to thank CLIF, this new climate and fungi group here at Duke, for their support, as well as the NIH. And with that, thank you so much for your time and this opportunity to present here today. And I'm looking forward to some questions and some engagement. So thank you very much.
Okay, well, thank you, Erica, and thank you, Andy, for two very, very exciting and beautifully presented talks. I just want to remind everybody that please do pop your questions into the Q&A function, not the chat function, the Q&A function, and we'll do our best to get to everything that we get. And I'm going to start the questions, and I have a question for you, Andy, and somebody who's observing that having made these insights to understand the role of copper in virulence and adaptation of pathogenic fungi, are there translational opportunities to take advantage of this? You began to talk a bit about this, but what can we do either in combination with other drugs or taking advantage of the knowledge of copper metabolism to perhaps do better in controlling fungal infections?
Thank you for that question. That really is a central a thought that we have all the time. One of the ways it has been explored in the past by some of our collaborators, Kathy France and then Ricky Festa, they looked at these molecules called copper ionophores that actually enhance the copper bombardment of cells and tried to use those as antifungal agents. Now, the caveat would be that these would be broadly cytotoxic for both mammalian and fungal cells, but they were able to chemically create these prodrugs that would hopefully be preferentially activated within fungal cells. And then also looking at fungal delivery systems, and they did have significant antifungal activity, but this differentiation between mammalian toxicity and microbial toxicity has yet to really be cracked with those compounds. However, in all of these, there are many fungal specific features of what I was talking about. These transcription factors are quite fungal specific. And that's why Dr. Washington is looking at the structure, trying to figure out, are there ways to perhaps inhibit the DNA binding activity of these transcription factors in a unique way? Or is there some other interesting aspect of them? The transcription factors give us insight into many different fungal specific copper relevant processes. And so that's the other eye that we're always looking for. And so, for instance, the cell1 is an enzyme that is, quite microbe specific. And, could that be, inhibiting an enzyme might be easier than these larger copper processes.
Thanks very much, Andy. I'll pass over now to Christina.
Erica, we have a question from someone who is interested in the TSL1 gene. I don't know if you mentioned it, but I guess this is part of a complex with the genes that you've talked about. The question is anything known about the role of TSN, part of the enzyme complex? And if I could add a second part, just from your lens as a structural biochemist, can you also just talk to us about how your structure of one component could be, how you think about that as part of that one protein as part of a complex.
Yes, thanks. Thank you for that question, both of those questions. So, right, so TSL1 is a part of a complex or part of the trehalose biosynthesis pathway in Saccharomyces cerevisiae. So today I talked about TPS1 and TPS2. There's also a third protein in Candida albicans. There's a third protein, TPS3. And TPS3 is a protein that lacks any known enzymatic activity. It has an N-terminal domain that looks like TPS1, except it lacks substrate binding residues. And then it has a C-terminal domain that looks like TPS2, the phosphatase domain, but it lacks the catalytic residues that are required for that activity. So the hypothesis, and it's been pretty well shown in Saccharomyces cerevisiae, that TPS3 and then also TSL1, which is the Saccharomyces has two proteins that lack activity, so TPS3 and TSL1, that those are involved in a complex formation. And my hypothesis about complex formation among these trehalose biosynthesis enzymes is that TPS1 forms this homotetramer that we've been able to show, and that these subunits can be exchanged, or these promers in that homotetramer can be exchanged, perhaps for the N-terminus of TPS2, which is TPS1-like. So I think this is a very dynamic process that we're not, showing when I show the cartoons. And that's something we're really excited to look into in the future, is how the subunits between TPS1 and TPS2, and also potentially TPS3 and TSL1, can affect complex formation and make it more efficient for there to be trehalose biosynthesis. And we really do think, in addition to the work done with TSL1 and Saccharomyces cerevisiae, we do think that these proteins form a complex. And the homotetramer of TPS1, I felt like was really supportive of indicating that these proteins can form a complex, and that we just need to find the right conditions and to be able to demonstrate and see if we can see an interaction between TPS1 and TPS2 in cells. We have preliminary data that shows in a bacterial 2 hybrid assay an interaction between TPS1 and TPS2, as well as TPS3, which is the non-catalytic form. So for one of the reasons that these proteins might form a complex is because T6P, which is the intermediate and in between TPS1 and TPS2, T6P is cytotoxic at high concentrations. And so one of the things that the cell probably would like to do is to quickly get rid of T6P as much as it can, to prevent it from accumulating, and also to keep it in the core center in this localization. So the more quickly TPS1 can make T6P, it's going to be much more efficient, and the cell's going to be much more healthy if T6P can be rapidly converted to trehalose. So yeah, does that?
Yeah, that's great. Thank you so much.
Okay.
Back to you, Neil.
Andy, if you don't mind, I'm going to slip in one of my questions before going back to the questions from the audience. So I was very interested in the part of the presentation where you showed that chitosan was an avid copper binder, and that if you, very beautifully, if you deleted the chitin deacetylases 1, 2, 3, or the CHS3 chitin synthase, which makes the chitin which is deacetylated by those deacetylases, that these cells became hypersusceptible to copper, presumably due to this mismethylation of proteins which get which are inside the cell. But my question really is, what's the consequence to the activity of those enzymes in the bound and non-bound form? Does that affect the properties of the wall as well as the secondary effects, perhaps, of the increased elevated copper inside the cell?
That's a great question. You're asking whether the presence or absence of the proteins actually affects the structure, affects the copper binding. within the, and that's one of the reasons that we did the two different types of mutants, because we were wondering what would happen if we got rid of 3 chitin deacetylases. But
Yeah.
I don't know that we can address that necessarily. Does removing the proteins, does that actually have an effect on the copper binding within the cell wall itself, or is it just the presence or absence of chitosan, within the cell wall? But I favor the latter, but I can't exclude the former.
Yeah. Yeah. That's interesting. Okay, well, I'll hand the baton back over to Christina.
Erica, our next question is from Manu de Riker. Thank you for a great presentation. If I saw this right, several of your analogs activated TPS1 rather than inhibited. Can you provide an explanation for this?
Yes, that is definitely true. That was what was shown on that slide. And I think that there's one explanation for the compound 2 increasing activity of TPS1 could be that it's binding and it's binding in the substrate binding pocket, but it also might be stabilizing TPS1. So that's one thing that can be happening. One thing I didn't show on that slide, and thank you for your question, and now I'll have to update it, is several of the compounds also were not soluble, and so they crashed out. And so when we ended up with 4456 was one of the ones that was not soluble, was soluble, sorry, and did not crash out and still inhibited the activity. But yeah, for the ones that are binding and increasing the activity of TPS1, my first thought is that they're increasing the stability. And it's an important thing to think about as well, because what happens when you increase the activity of TPS1 in a cell? Do you make too much T6P? Does that then accumulate, and is that now toxic? And so it is probably worth going back and kind of looking at some of those compounds, and they're in the freezer. So thank you for that question.
Yeah, what you're saying draws some parallels for me of ergosterol pathway and what happens there. So
yes, exactly.
Back to you, Neil.
Thanks, Christina. So Andy, Cheney Ramos has got a question. Loves your talk. Right at the beginning of the question, the patient that you mentioned, the farmer, she's said the question is the treatment of the patient you mentioned at the beginning when which was cryptococcus, and when the lesions form, the standard treatment is to remove surgically the lesion, perhaps along with antifungal treatment. But what do you think might have caused these lesions to form so readily in that patient? Do you have any ideas about susceptibility?
Yeah, so typically we would not perform surgery in a patient like this. We would treat the patient with, antifungals, and a certain, number of people, the lesion would get better, obviously with some scarring there. But the question is, what might have caused these types of lesions to form? And perhaps Christina can tell us more about Cryptococcus gattii and the genomic differences, that may favor cell wall differences that you, Neil, can talk about. But I really think that what is happening here is that there are certain microbial features that favor a very different immune reaction within the host. So instead of forming a diffuse meningoencephalitis, from a small number of fungi dispersed throughout the leptomeninges, we have a focus of Cryptococcus that has grown and is inducing a larger cryptococoma or focal lesion. And I believe there are likely some cell surface changes, difference in the antigenicity or the types of immune response that are being induced by these different strains. And we have no idea what happened in this one strain. But could it have been a gattii? Could it have been a variant neoformans that had a variant cell surface structure? That would be my thought. The one sort of sad note about this patient, although he survived his Cryptococcus infection, he didn't survive the subsequent lung cancer that he did develop from his years of smoking. But again, he had several years of really effective cure of this infection.
Okay, thanks, Andy. And then back over to you, Christina.
Erica, we have a question from William Curiso. Erica, beautiful research and a great talk. This is a tough one. Can you speculate on if there's functions for these unstructured domains? Yeah, it's you've already got this very good structure, but you talked about wanting to get those correct.
Yeah, so thank you for the question. We have looked for a function for these intrinsically disordered domains. And one of the reasons, and due to time, I didn't go into this, but one of the reasons we're particularly interested in this domain is because we find this domain in cryptococcal species and closely related basidiomycete species. But when we look at TPS1 sequences outside of those fungi, and ascomycetes and mucor, we don't see this intrinsically disordered domain. So it seems like it's something that's specific to the basidiomycetes that must be important. The other reason we're really interested in this intrinsically disordered domain is that in the fungi that do have the domain, it's highly conserved. So greater than 95% conservation of the residues in that domain in the fungi that have the domain. So not only is it basidiomycetes specific, but it's pretty conserved within those basidiomycetes. So we think it must be doing something. So our first idea to check was, is it involved in thermotolerance? It's not involved in thermotolerance. It's not required for thermotolerance. We also measured in vitro, is it required for activity? So we made a chimera where we replaced that entire 92 residue loop with 6 amino acids from Candida albicans TPS1. And we were able to purify this protein and folded nicely, similar to the Cryptococcus neoformans TPS1. And then we measured its activity. That's the same activity as wild type. So it's not required for activity. We also looked for, is it required for osmotic stress responses? No. So our next hypothesis that we're really excited about is to see if this intrinsically disordered domain is involved in forming condensates. And so we have a collaboration ongoing now with Amy Gladfelter, also here at Duke, where we're looking at condensate formation. And is this intrinsically disordered domain involved in the formation of molecular dynamic complexes, condensates inside of the cells? Another hypothesis is that it's a surface for protein-protein interactions. And so one thing that we might be able to attempt is if we can find an interaction that stabilizes the intrinsically disordered domain, maybe we could solve that structure and get an idea of if it's important for mediating an interaction with another protein. But that is something we are looking into, and we're really excited to see what it does because I think it's going to be important. So thank you for your question.
You're saying that this is one region that's different from Candida albicans. Was this one of the major regions? You must have compared the structure you found to Candida albicans?
Yes, so I did. I compared the structure, and so TPS1 has those two subdomains. I'm backwards to you guys, but so the N-terminal and then the C-terminal. The loop, the intrinsically disordered domain actually forms a loop. So where it comes off of the structure is right next to where it comes back. And that's how we were able to make the chimera with the TPS1 enzyme. And so it's similar with the Aspergillus fumigatus, doesn't have the loop, but when you compare the structures, the loop just goes out and comes right back in, right in the same region. Is also why it was easier kind of to delete that part of the protein. So it's coming off of the N-terminus, but the rest of the structure remains unperturbed. So it's not even near the catalytic pocket. So we were wondering if there's some sort of allosteric regulation that would affect the catalytic pocket, but deleting it didn't affect the activity. So.
Okay.
Neil,
Okay,
another question.
Andy, if I may ask a question, your emerging model shows, chitosan to be like a copper barrier in the wall, and I wonder if there's a biological question of addressing that. So, for example, Candida's got very small amounts of chitin in its wall. It does have a deacetylase, whereas the mucorrhizis fungi like Mucor and Rhizopus have got a lot of chitosan in their walls. And I wondered whether you'd thought, well, maybe I could test this model broadly across the fungal pathogens by doing CDA deletions in chitosan rich and chitosan poor organisms and see whether that affects their susceptibility to medium copper.
Yeah, that's a brilliant idea. You know, I hadn't really thought about that. And I tend to be too Cryptococcus centric, and I really love the idea of looking across fungi with different levels of cell wall sugars and the content of their cell walls. You know, and then I think your point as well before is it the chitosan itself, which we know binds copper, or could it be something enriched in the chitosan layer? You know, different proteins. We don't yet know. But I do think that our data would suggest that like that CBI1 does bind copper, and it's likely present at least in or near this layer. And so there's likely a protein polysaccharide interaction occurring. But how that might relate to copper resistance with Candida versus the mucorales and others is a great question.
Yeah. Well, it's a nice, clear kind of hypothesis you've set up, which would be nice to test.
Yeah, I love it.
So I'm going to pass back, but I may have mentioned, Christina, that if there is time, I have a question also for Erica.
Okay, I had one last one, maybe quickly. Erica, I was curious with your 4456 inhibitor. Had you tested, because you know kind of where it directly binds, potential resistance, if it, if you know kind of where it's binding, if you change those positions and then kind of that would be maybe a mechanism of resistance. Are you only affecting resistance to that drug, or are you also affecting the way the products are binding and killing the enzyme essentially?
Right. Thank you. Thank you for that question. Unfortunately, based off of where 4456 is binding in that substrate binding pocket and its proximity to those residues that I showed you on the slide, those residues are also required for function. So if we mutate those residues and show 4456 doesn't have activity, well, that's because TPS1 doesn't have activity with those residues. So that's something we're interested in doing, and it would have to be something that happens in the next probably library of derivatives, where we're interested in building out from the substrate binding pocket to contact different parts of TPS1 to make this a bigger, more active molecule. So that's something that we are interested in. We have set up some drug resistance assays to see if we can get suppressors of TPS1 of 4456 activity. So far, we have not been able to get any colonies, but the point being to figure out what are the residues that are required for the activity of 4456 and TPS1. So while it's good that so far we don't have those hits, perhaps we will get them, and we'll keep trying, and that'll give us an answer about more information about the mechanism of 4456.
Great, thank you.
Yeah.
Okay, well, I think we're actually out of time, and we've always promised we try and keep things on time so you can get away at the right time. So I'm going to end by thanking both Andy and Erica for fabulous talks, beautifully presented. And also just to remind you that we have a monthly meeting. The next one is on July the 24th. That's Jamie Canopka and Jane Usher. So please do join us there. And let me thank Andy and Erica on your behalf, because you can't hear the sort of deafening applause which is going around the world at this moment because of our IT setup, but you can be sure it's there. So thanks very much indeed. And thank you also to Christina for partnering with me, and all the people who set up MycoTalks for their work in putting together the program. So thanks again, and look forward to seeing you in a month's time.
Thank you.
Bye-bye.
Bye.